Hey guys, my name is Max and welcome to Simple Biology. Today we're going to be talking about membranes. Now, if you've ever studied biology or even seen a diagram of a cell, you know that the cell membrane is one of the most prominent features of any cell. In addition to the cell membrane, most cells have other membranes inside of the cell itself. And almost all of the organelles that we talk about in this unit have one or more membranes involved directly in their function. Because of this, it's very important to understand membranes and how they work. Now, the first thing we're going to look at with a membrane is its structure. Now, a membrane is made up of a bunch of phospholipids. A phospholipid looks a little bit like this, and it consists of two fatty acids, which are these two chains right here, all the way up to the oxygen atoms, as well as a glycerol right here, and a phosphate on this end right here. Now, the interesting thing about a phospholipid is that it has two regions with very distinct chemical properties. Namely, this blue region is very nonpolar, and as a result is hydrophobic, and this orange region is polar and even charged, making it very hydrophilic. As a result, when you get a bunch of these phospholipids together, they arrange automatically in a double layer with the tails on their inside. This double layer, is also called a phospholipid bilayer. The reason the tails automatically go on the inside is because hydrophobic and hydrophilic molecules, do not mix well. Because of this arrangement, you have this whole region of hydrophobic parts of molecules, and it's completely separated from a whole area of hydrophilic molecules and parts of the molecules. Now, in addition to all these phospholipids, membranes are also scattered with proteins. It's not shown in this diagram, but membrane proteins are very important to the functions of membranes, and we're going to be looking at that later in this video. Now, the next property of membranes that we're going to look at is fluidity. In other words, how quickly or how easily a membrane will move. Now, there are several factors that influence fluidity, but the first is temperature. Fluidity increases directly with temperature, meaning that as the membrane gets hotter, as its temperature increases, these phospholipids will move around more, and the membrane as a whole will become more fluid. If you remember from chemistry, temperature is a measure of how quickly things move, so this makes sense. On the other hand, at low temperatures, those phospholipids will move around less, and the fluidity will decrease. Now, there's a very important molecule in membranes that impacts fluidity, and it's called cholesterol. Now, cholesterol is very important because it opposes the changes in fluidity due to temperature. Now, if you ever read the side of a cereal box, you've probably heard of cholesterol, and it's not as bad in the right proportions, in the right amounts, as most health professionals seem to make it. Because of this role in fluidity, it's entirely vital in most membranes. Now, I'm going to explain why it functions like this. As the temperature increases, like I said, the phospholipids will move around more. The cholesterol will prevent them from moving around quite as fast because as they start to move around more, they'll hit the cholesterols. They'll rub against the cholesterols, and that will prevent them from making the membrane as fluid as it would be otherwise. On the other hand, when the temperature decreases, those phospholipids have the tendency to clump together and pack very tightly, and the cholesterol will get in the way of that and prevent it from happening. Now, the last thing with fluidity is that we're going to look at the difference between unsaturated lipids and saturated lipids. I'm going to bring you back to this other diagram, and this phospholipid actually has two different lipid chains. All phospholipids have two chains. In this case, one is saturated and the other is unsaturated. The difference is that an unsaturated lipid chain or hydrocarbon chain will always have one or more double bond, as you can see right here. Now, this influences fluidity because unsaturated lipids are much more fluid than saturated lipids. This is because since unsaturated lipids have that double bond, they don't pack down quite as well as saturated lipids. And in addition, the lack of a double bond, the absence of a double bond in saturated lipids, makes the chain much more rigid than it would otherwise be. Now, like I mentioned earlier, almost all membranes have proteins on them. These proteins are called membrane proteins. Now, the important property that distinguishes a membrane protein from any other protein is that it is amphipathic. Now, it sounds more complicated than it is. Amphipathic basically means that it has a hydrophobic region of the protein and a hydrophilic region of the protein. Another great example of an amphipathic molecule is a phospholipid, like we mentioned earlier, because it has those two distinct regions. Now, membrane proteins have a large variety of functions, but some of the biggest ones are these four that I'm going to address. First is catalysis, meaning that the protein can function as an enzyme, in which case it will allow a chemical reaction to occur more quickly. The next one is signaling. Between two cells, they sometimes communicate through signals, and those membrane proteins are very important in that. And as another type of signaling, sometimes those cells will need to recognize each other, and so cell recognition also relies on membrane proteins. The last one is transport of different materials into and out of a cell, which nearly always rely on membrane proteins. Now, the last thing with membrane proteins is that sidedness or orientation of a membrane protein is very important. As you can see in this diagram, one side of the protein has this little blob-looking thing, and the other side does not. And that blob-looking thing is most likely very important to its function, and so it has to be present on one side in specific of the membrane. The interesting thing is that sidedness always depends on the orientation in the endoplasmic reticulum and in the Golgi body. Again, looking at this diagram, you can see that when the blobby-looking thing is on the inside of the Golgi body, in the red region, it'll end up on the inside of the vesicle and on the outside of the cell membrane. On the other hand, if it was on the outside of the Golgi body, in the blue region, it would end up on the inside of the cell membrane, again in the blue region. This is kind of a good diagram to look at when you're thinking about that. Now, there are two very important processes, chemical processes that function a lot in biology, and they emerge with greater effect when you look at membranes. The first is diffusion, and diffusion is a natural process that occurs when there are unequal concentrations of a substance of any kind. Now, looking at this diagram right here, you can see that the particle, most likely a solute in a solution, is present in abundance, is very concentrated in this region, whereas it is not as concentrated in this other region. As a result, because of diffusion, that solute will naturally diffuse from an area of high concentration to an area of low concentration. Now, the problem comes when you have a certain type of membrane called a semi-permeable membrane. In this case, this membrane is permeable to water, but not to the solute. In this case, you have a process called osmosis, which is basically the diffusion of water across the membrane. It's a little more complicated than that, so we're going to look into it with greater detail. This is what the water levels would be if gravity were the strongest force playing a role. Obviously, the upper levels of the water are at the same level because of gravity. In this side of this membrane, there are more solute particles than in the other side. As a result, this side has a higher solute concentration than this side. over time, the solution will actually settle a little bit more like this, opposing the effect of gravity. The reason this will happen is because the solute cannot travel across the membrane, water will instead travel across in order to equalize that difference in concentration. And so as you can see in this diagram, the two sides have a very equal concentration, but not the same water level because of osmosis. Now, the last thing we're going to look at with membranes, and membrane proteins in particular, is transport proteins, like I mentioned earlier. Now, there are two types of transport when we talk about transport proteins. There's passive transport, and there's active transport. We'll be talking about the distinction in a little bit, but basically passive transport, which we're going to start with, is diffusion that uses proteins. Now let's back up a little bit. Why may the solute, the particle, need proteins in order to cross the membrane? Now, from chemistry, we know that if the particle has certain characteristics, it will be able to cross the membrane on its own. If it's small, and if it's nonpolar. For example, oxygen and carbon dioxide can very easily cross membranes. On the other hand, if it's charged or if it's too large, it will need some sort of protein in order to transport it across, and that's why we need transport proteins. So these show two different transport proteins. Both can undergo passive transport, but the difference is that this is a channel protein, whereas this is a carrier protein. The difference is that the carrier protein will allow for facilitated diffusion, whereas the channel protein will not. It will allow the protein to travel straight through. Now, facilitated diffusion is just where this particle will go into the specified spot, and that will force the protein to change shape, allowing the particle to exit on the other side. So obviously, the facilitated diffusion has a little bit more control over the diffusion than the channel protein will. And this is where active transport comes in. Active transport only occurs with these carrier proteins, and it uses energy. Now, the reason it uses energy is because it actually opposes diffusion. What active transport will do is it'll take a particle that with very low concentration and move it to an area with a very high concentration. This opposes the effects of diffusion, and that's why it uses energy. Now, the reason you can't have a channel protein use active transport is because it doesn't have that control over it. So you can see that a lot of this is kind of instinctive, and membranes are really just as simple as that.